Good morning, everyone. Thank you all for being here. Today I'm going to talk through AI-assisted clinical data acquisition and interpretation. And we are doing this in the context of sarcopenia, which, as you know, is a age-related pathology, common and ruthless in most mammals. There are no therapeutics currently approved, and the companies in the space are moving pretty slowly, so we kind of had our own go at this. Hope you can see these pretty well with the sun shining. These are some of the participants in the clinical trials. As you can see, they are pretty frail, very thin, significant muscular dystrophy present across. They're all old cats, 10 years or more. And we've tried to incorporate AI at multiple steps in our protocols. We couldn't actually replace lawyers. We did some paralegal AI agents. But here we're showing the translated approval from the state authorities to run the clinical trials under European law. And the plan was pretty simple. So we have a vast network of veterinarians that we work with, and from the patients that they see, they recruit the cats which fit certain inclusion criteria, like a certain body mass index, body condition score, age, and stuff like that. And then after that, we take a baseline measurement. We are administering our own therapeutics, and then we compare post-treatment in blood work panels and also physiologically. We actually have a pretty nice method that can isolate DNA, RNA, and proteins from the same sample. So we can have like a big picture with everything that's happening in the organism. For the purpose of this talk, however, we're only going to be looking at RNA. You might be familiar with the nanopore sequencing technology. So this is where we have these nanopores and the nucleotides are passing through. There's a change in the electrical current. And then using machine learning algorithms, that perturbation of the current is translated into base sequence. This is our PromethION running. This generates quite a lot of data. We stopped the runs at about 200 gigabytes per sample. This was because we're doing direct RNA sequencing, so we're not doing any reverse transcription because we want to see molecule modifications as well. As you might be familiar with epigenetics, you get the same type of modifications on RNA molecules as well that do impact stuff like later on. We're able to detect those modifications with the changes in the electrical current here. And this is where machine learning comes into play. We have a few different models that we can use with different levels of accuracy. And you can see on the right-hand side, there's a graph of an alignment in the CD52 gene. And we can see when we align with the fast accuracy model, we get a reasonable number of reads, but the variability in spurious variants is pretty high, which isn't necessarily true to the biological context. So we see the change here quite significant when we move to higher accuracy models, where we're actually able to align more reads to the same transcript, and the number of spurious variants decreases, as you can see on the top there. So this just gives us more confidence in the data that we're looking at, and we're using computational credits here. We have raised our pre-seed round last September, and we have received about 350K in credits for Azure and AWS and Google AI and stuff like that to process these big data sets and understand what's happening pre- and post-treatment. Here's a sneak peek at the data. We're showing control in red and post-treatment in blue. So first, we've looked at a few markers. We're showing CD19 here, which is associated with the number of B cells. The number of B cells is shown to decrease in aging, and we observe here what could indicate immune rejuvenation post-treatment. But you can see this as well if the organism is infected or is just having an immune response to something, an adaptive immune response. So we also looked at CD69 to kind of measure the chronic level of inflammation. We were super happy to see that this is significantly decreased post-treatment. Again, further corroborating our immune rejuvenation. And we dived in even further, so we looked at heat shock proteins. As you might be familiar, these appear in contexts of calorie restriction and sauna, both mechanisms that and interventions that are associated with longevity. So we were really happy to see multiple subsets of these going up. So of course, both me and my co-founder Alex, who you see on stage after this, are molecular nerds, and we love this data and to dive in numbers. But what hyped us up the most was actually clinician observation. Where the veterinarians that we worked with were able to actually see an increase in muscle mass and also in the overall activity of the cats, their presence, they're just being more active. And this was just over a couple of weeks. This is just interim clinical data here. We are filing this for a patent, and we'll be sharing more once that's done. So that was pretty much it for my talk. I'm happy to take any questions. Thank you.
Questions?
Okay. I was just wondering, what RNA modifications specifically are you interested in? Like I said, I think M6A is one of them, for instance. And what was the desire to look at that originally, and have you found anything interesting with those modifications at this point?
Yeah, so we are still processing the data. We are looking at multiple modifications. So you do have the M6A, but there are different types of methylations, and then you have the adenosine to inosine, which influences stuff. So similarly, like you'd have CPG islands, where we're looking at that across the whole transcriptome. But we are still processing that. Thank you.